Here are all of my materials. I forgot to say that today is September 21st and this is the Differential Staining Lab. My name is Danae Hall and right now I am just smearing the teeth cells onto the first slide. Now I'm going to allow the heat fixed slide to cool for 10 minutes. So my first slide is done cooling, so I am going to add a drop of the Crystal Violet Gram Stain number 1 to the center of the drawn circle on my first slide. I'm going to allow the slide with the Crystal Violet Gram Stain on it to sit and dry for about 60 seconds. Alright, so now that the gram stain has dried, I'm just going to rinse it off with some water. Alright, now I'm going to look at my gram stained first slide underneath the microscope. This is the first slide under low power magnification. This is the first slide under medium magnification. This is the first slide under high power magnification. Now I'm going to take a single drop of the red Congo dye and put it right in the middle of the slide that I just labeled. And I'm just going to scrape the inside of my mouth again. And now with that swab, I'm just going to agitate the red Congo dye to get as many cells as I can on that slide. So I'm going to just wipe this dye across the rest. Now I'm going to look at the second slide with the Congo red stain, which is fully dried under low, medium, and high power magnification. The red Congo stained slide under low power magnification. Red Congo stained slide under medium power magnification. Red Congo stained slide under high power magnification. I have my nutrient broths and my slides are prepared, and then I have my inoculation loops sterilizing in alcohol. Alright, so I have my inoculation loop. I'm just going to shake it dry. E. coli nutrient broth tube. I'm going to pass this through the flame, and I'm going to insert the loop into the broth and then transfer the liquid onto the middle of the circle on my E. coli. Now I'm going to flame the lid again one more time. I'm going to put the inoculation loop into bleach before disposing of it. Next is the S. cerevisiae, and then pass the lip through the flame to sterilize it. And I'm going to take my dry inoculation loop, insert it into the tube, and transfer the liquid onto the S. cerevisiae slide. Pass this over. Now that all my sides have been heat fixed, I am going to allow them to cool before we do the staining. Alright, so I have my E. coli slide. It's all cooled and it's in the staining tray, so I'm going to add a drop or two of Crystal Violet. I'll allow this to sit for about 60 seconds. The slide has been sitting for about 60 seconds. Rinse it off with some tap water. The next step is going to be to put 2 to 3 drops of this PVP iodine. Now that the iodine is rinsed off, the next step is to put 2 to 3 drops of decolorizer on the stain. It's been about 30 seconds, so I'm just going to rinse the decolorizer off. Add 2 to 3 drops of safranin on top of the stain. Alright, it has been about 30 seconds, so I'm just going to rinse off the safranin, and now I'm going to allow my stained slide to just dry on some paper towels. I'm going to look at it under the low, medium, and high power magnifications. So this is the stain of the E. coli slide under the low power magnification. This is the E. coli slide under the medium power magnification. E. coli slide under the highest power magnification. So my next slide to stain is the S. cerevisiae slide. So the first step, or 1 to 2 drops of Crystal Violet. Alright, so it's been about 60 seconds, so I'm going to go ahead and rinse off Crystal Violet. So the next step is to add 2 to 3 drops of the PVP iodine. Alright, it's been about 60 seconds for the S. cerevisiae slide, so I'm just going to rinse it off with some water. The next step is to add 2 to 3 drops of decolorizer. I am going to rinse this off, and the next step is to add 2 to 3 drops of saffron in. Alright, so for the S Cervicé, it's been about 30 seconds with the saffron in, so I'm just going to rinse it off, allow the S Cervicé to completely dry on some paper towels. So now the S Cervicé slide is dry, so I'm going to place it on the microscope stage. S Cervicé slide under low power magnification. The S Cervicé slide under medium power magnification. The S Cervicé slide under high power magnification. My next slide is the S Epidermis slide. So my last slide to view is the S Epidermis slide. S Epidermis slide under low power magnification. S Epidermis slide under medium magnification. The S Epidermis slide under high power magnification. So that is the end of the differential lab. So I'm going to soak all of the reusable tools in bleach, clean up my station, and then return all of the cultures